Okay, good afternoon, children. Today in our class, we are going to discuss about eukaryotic cell. Right. Eukaryotic cell. Here, generally the eukaryotic cell includes protista, fungi, plantae, and animals. All those are belongs to eukaryotic cell. Here, what is the main difference between the prokaryotic cell and eukaryotic cell? Here, the prokaryotic cell does not contain well-defined nucleus. Well-defined nucleus means the nucleus not surrounded by nuclear envelope. So, well-defined nucleus and all cell organelles are absent in prokaryotic cell, except 70s type of ribosomes. Apart from prokaryotic cell, the eukaryotic cell consisting with the well-developed nucleus and all cell organelles, membrane-bound cell organelles and non-membrane-bound cell organelles. All those are present in the eukaryotic cell. Here, generally, the eukaryotic cell includes, we are already told, protista, fungi, plantae, and animalia. Here in our previous class, we discussed about plant cell, differences between plant cell and animal cell. Here presence of cell wall is unique property of plant cell which is absent in animal cell. Here the plant cell contains chloroplast which is absent in animal cell. Sap vacuoles are present in plant cell but it is absent in animal cell. Right. Centriole is non-membranous cell organelle which is characteristic feature of animal cell but not characteristic feature of plant cell. Now here, now I would like to ask one question. A and B are two non-membrane bound cell organelle, non-membranous cell organelles, but A is present in plants and animals, B is present in only animals. Here find the A and B respectively. A and B both are non-membranous cell organelles. A is present in both plants and animals, but B is present in only animals. What are the A and B respectively? A is ribosomes and B is centriole. Okay, right. Now here in the eukaryotic cell, now we are going to discuss about cell wall. Right. First of all, cell wall. Cell wall is unique property of plant cell, but not only the plant cell, bacteria cell contains cell wall and also fungus also contains cell wall. So presence of cell wall is not only the characteristic feature of plant cell, it is also seen in fungi and bacteria also. Right. Now we are going to discuss about plant cell wall. Here, who discovers the cell wall first? Robert Hooke discovers cell wall. Here, he discovers cell wall from cork cells in which plant means oak tree. Here it is called Quercus cuber. Quercus suber, nothing but oak plant. Robert Hooke first discovers cell wall. Cell wall. He discovers cell wall in cork cells of oak plant. Here cork is a living tissue or non-living tissue means cork is non-living tissue. Right. He discovers cell walls in cork cells. So that's why cell wall is also non-living. Cell wall is also non-living. Here the cell wall is formed from protoplasm. Cell wall is formed from protoplasm. Right. The protoplasm includes cytoplasm and nucleus. Cytoplasm and nucleus collectively called as protoplasm. Right, Amma? So the cell wall is synthesized from protoplasm. Here generally in plants, the cell wall is consisting with three layers. First one is middle lamella, second one is primary cell wall, then secondary cell wall. Generally these are the three wall layers that were present in the plant cell. Here middle lamella, primary cell wall and secondary cell wall. Generally in immature plant cells, nothing but young plant cells. Example of young plant cells, nothing but meristematic cells. Here the meristematic cells contains only two wall layers. Young plant cells are immature cells. Immature cells or young plant cells contains only two wall layers. What are they? Means middle lamella and primary cell wall. But here all these three wall layers are found in mature plant cells. Okay, I think you got it the information here. Middle lamella, primary cell wall and secondary cell wall. Generally these are the three wall layers of the plant cell. Here middle lamella and primary cell wall, only two wall layers are present in the young plant cells or immature cells. Mature plant cells contains three wall layers. Now let us see about individual wall layers. Here middle lamella, first of all middle lamella. Here just it is middle lamella. Here it middle lamella, it is first formed wall layer. Here the middle lamella functions as cemented layer between two adjacent cells. Means here just this is the middle lamella. The middle lamella joins the two adjacent cells. That's why it is called as the intercellular matrix. Middle lamella considered as intercellular matrix because it joins the two adjacent plant cells like a cemented layer. Hence it is called as the intercellular matrix. Intercellular. And middle lamella generally it is first formed wall layer. First formed wall layer is middle lamella. Generally, the middle lamella is formed during the cytokinesis of cell division. Right? It is developed from cell plate. Cell plate. Here, the cell plate undergoes into various modifications. Various modifications then form middle lamella. So, cell plate is precursor of middle lamella. Right? Here, generally the middle lamella composed of calcium pectate and magnesium pectate. Calcium pectate and magnesium pectate. Okay, right. Here, which cell organelle plays an important role in the formation of middle lamella means Golgi apparatus. Golgi apparatus plays an important role in the formation of cell plate. The cell plate undergoes into various physical and chemical changes then form middle lamella. Right? The middle lamella is glue or cemented layer between two adjacent cells. Here, in a multicellular organisms, the two cells are get separated from each other due to dissolving of middle lamella. Just what I said, Middle lamella functions as a glue. Middle lamella functions as a cement, means which joins the two cells. If middle lamella get dissolved, what will be happen? The two cells get separated from each other. So here actually in middle lamella is get dissolved during ripening of fruit. Ripening of fruit nothing but maturation stage. Okay, here the maturated cells get separated from each other due to dissolving of middle lamella. So that's why what I am saying here, the middle lamella get dissolved during ripening of fruit. Okay, right. This is middle lamella. Second one is primary cell wall. Primary cell wall. Here, generally, the primary cell wall is present inner to middle lamella. Here, just this is primary cell wall. It is present inner to middle lamella. Here, generally, the primary cell wall is thin and elastic nature. Primary cell wall is generally thin and elastic in nature. Here the primary cell wall is, what is the function of primary cell wall? It is meant for cell elongation. Cell elongation or cell expansion. Here generally the primary cell wall contains a matrix which is composed of pectin. So pectinous matrix is present in the primary cell wall, pectinous matrix. Here the cellulosic microfibrils are enters into the pectinous matrix, then form a network like shape. Okay, right. Here some areas are thinner but not this contains with the cell wall material. So the thinner areas of primary cell wall are called as the primary pit fields. Primary pit fields. Primary pit fields are presence of characteristic feature of primary cell wall. So the thinner areas and means thinner areas means the primary cell wall, in the primary cell wall where the cell wall depositions are absent, such is called as the thinner areas. The thinner areas of primary cell wall called as the primary pit fields. Okay, here what is the use of primary pit fields? Here generally the cytoplasm of two adjacent cells are connected through primary pit fields. These cytoplasmic connections are called as the plasmadesmata. Plasmadesmata. So the plasmadesmata connections are established through the primary pit field. So the primary pit fields are meant for intercellular transportation of materials. Okay, right. Generally, the primary cell wall is composed of cellulose, hemicellulose, and pectin. Here, what are the materials? What are the primary cell wall materials? The primary cell wall materials are cellulose, hemicellulose, and pectin. All these are the primary cell wall materials. Now let us see about the secondary cell wall. Here, what is the secondary cell wall? When it is formed means when the cell becomes maturated, when the cell becomes mature, or when the cell becomes developed. Here the primary cell wall becomes diminishes, means nothing but disappear, but not clearly fully disappear. Here during that time the protoplasm lays somewhat layers inner to primary wall. These are called as the secondary cell wall. Secondary cell wall. Okay, here just this is middle lamella. Here inner to middle lamella is called primary cell wall. Inner to primary cell wall is called as the secondary cell wall. Here inner to secondary cell wall, what is there? Means here just this is the plasma membrane which is present inner to secondary cell wall. That's why what I am saying, the secondary cell wall is a layer which is present between the plasma membrane and primary cell wall. Secondary cell wall is a layer which is present between the plasma membrane and primary cell wall. Right. Now let us see about the complete discussion about the secondary cell wall. Right. Secondary cell wall. Here at the time of maturation, the protoplast produces additional wall layers inner to primary cell wall, which is called as the secondary cell wall. Here in a secondary cell wall, the cellulose forms a matrix here in which the somewhat materials, those materials nothing but secondary cell wall materials such as lignin, suberin or enters into the cellulosic microfibrils, then increase the thickness of the secondary cell wall or increase the thickness of the wall layer. So what are the differences between the primary cell wall and secondary cell wall? Primary cell wall is thin and elastic in nature. It is responsible for cell growth, elongation or expansion. Right. So primary cell wall materials are cellulose, hemicellulose and pectin. These are about primary cell wall. Next, secondary cell wall. Secondary cell wall is thin and inelastic in nature. Secondary cell wall is thin and inelastic in nature. Here, secondary cell wall is composed of lignin, suberin and cutin. All these are the secondary cell wall materials. Next, what is the function of secondary cell wall? Secondary cell wall is responsible for increase the thickness of cell wall. Okay, here this is about the cell wall, whether it is middle lamella, primary cell wall and secondary cell wall. Okay, right. What are the secondary cell wall materials? Lignin, suberin and cutin. All these are the secondary cell wall materials. Generally, the secondary cell wall materials are found in tracheids. Tracheids composed of lignins. Endodermal cells, lignin and suberin. Cork cells composed of only suberin. Here, next, especially algal cells. The cell wall of algal cells composed of cellulose. Next, glucagons, mannans and galactans along with other minerals like calcium carbonate. All these are found in the cell wall of algal. The algal cell wall composed of cellulose, glucagon, malacton and galactons along with somewhat minerals like calcium carbonate. This is about cell wall. Now we are going to discuss about cell membrane or also called plasma membrane. Plasma membrane is a biological membrane here which is called selectively or differentially permeable membrane but not semi-permeable membrane. What is the difference between the selectively or differentially permeable membrane or uh, semi-permeable membrane? Here, what is the semi-permeable membrane? Semi-permeable membrane allows only solvents but not allows solutes. But plasma membrane is a selectively permeable membrane. Plasma membrane is selectively permeable membrane. What is the selectively permeable membrane? Or it is also called differentially permeable membrane. Selectively or differentially permeable membrane allows all solvents and some selected solutes, which is called as the selectively or differentially permeable membrane. Okay, how can you differentiate semi-permeable membrane and selectively permeable membrane? Semi-permeable and semi-permeable membrane allows only solvents but not solutes. Selectively permeable membrane allow all solvents and selected solutes only. Now we are going to discuss about plasma membrane. Plasma membrane. Right. Here the plasma membrane is called plasma lemma. and it is a lipoproteinaceous membrane. It is selectively or differentially permeable membrane. Here, the studies on plasma membrane, first of all, carried out on RBC of human beings. The studies of plasma membrane is first of all carried out on RBC of human beings. Okay, here what are the components? Here what are the components of plasma membrane? Generally, the plasma membrane is composed of proteins and lipids. Here generally the proteins in the plasma membrane are three types. Number one, structural proteins. Second one is carrier proteins. Third one is catalytic proteins. Generally the proteins that were present in the plasma membrane are three types of proteins are there. Structural proteins, carrier proteins and catalytic proteins. Here what are the carrier proteins? Porins and pumps. These are the carrier proteins. Here, what about the catalytic proteins? Catalytic protein nothing but enzymes. The protein with catalytic property is called as the enzymes. Okay, right. Lipids. Here, generally, the plasma membrane contains glycolipid and phospholipids. Phospholipids. What is glycolipid? Here the lipid molecule which is associated with glucose is called as the glycolipid. So the proteins and lipids, these are the two components of the plasma membrane. Okay, here what is the arrangement of proteins and lipids in the plasma membrane? Okay, here in the plasma membrane, each lipid has two parts. One is head, second one is tail. Head is polar, tail is non-polar. What is polar and what is non-polar? Polar means the head part is dissolved in the water. That's why it is called polar nature. What is non-polar? Here which is not dissolved in the pure solvent. What is the pure solvent? So these are not dissolved in the pure solvents which are dissolved in the benzene, alcohol, petroleum, ether. Now that is called as the non-polar nature. So the polar head, another one is the non-polar tail. Okay, here in the plasma membrane, the polar head is located towards outside and non-polar tail is located towards inside. Polar head is located towards, this is the polar head, it is located towards outside. Here it is non-polar, non-polar tail is located towards the inside. Polar head is outside and non-polar tail is located towards the outside. Here generally the lipids are composed of hydrogen and carbon also. That's why the lipids are a chain of hydrocarbons. Here why does the lipid? Here the tail of lipid is located inner side of the inner side, because here the non-polar tail of saturated hydrocarbons are protected from aqueous environment. Due to this reason, the non-polar tail is located towards the inside. Here, why does the non-polar tail is located towards inside? Because the non-polar tail is protected from aqueous environment, because such type of arrangement. Here, my question here, what is the ratio of head to tail in a lipid molecule? What is the ratio of head to tail in a lipid molecule? Head 1, tails are 2 tails. So the head to tail ratio is 1:2. It is very important question. This is about the lipids. Now second one is the proteins. Here the proteins, here based on the location and based on the ease of extraction, the proteins present in the plasma membrane is two types. What are the two types of proteins are there? Internal proteins and external proteins. Internal proteins nothing but intrinsic proteins, external proteins nothing but extrinsic proteins. Here the proteins proteins based on the location and based on the ease of extraction. The proteins in the plasma membrane are two types. Number one, integral or internal. Second one is the extrinsic proteins. Okay, here internal proteins, here just this is the integral protein molecule. Okay, here it is lipid molecule. The integral proteins are completely or partially embedded inside the lipid molecule. Okay, right, here which are not easily removable. Second one is the external. Here these external proteins are located on the surface, just the external surface. These are the external proteins. So the external proteins are located on the only external surface. These external proteins are easily removable. So based on the nature, the proteins are three types. Based on the location, the proteins in the plasma membrane are two types. Integral proteins are intrinsic proteins. Extrinsic proteins are external proteins. Easily removable proteins are extrinsic proteins. Here next, completely or partially embedded proteins are called as the internal proteins. Okay, here right, plasma membrane. Here different scientists, so many scientists propose their own individual models of plasma membrane. Okay, here first one is PLLP model, here which is called sandwich model, here which was proposed by Daniel Lee and Davison. Here this model is not acceptable. PLLP model of plasma membrane was not acceptable. Why? Because according to this model, the plasma membrane is solid in nature. That's why it cannot functions as selectively or semi-permeable nature. So that's why, however, it fails to explanation of selectively permeable nature of the plasma membrane. That's why it is not accepted. Second one is the unit membrane model. Various scientists proposed different models. Already told, Daniel Lee and Davison proposed PLLP model. What is PLLP model? PLLP model. Here just this is protein. Here this is also protein. Next here it is lipid. Here just this is the lipid. Here P, protein, lipid and protein. It was proposed by Daniel Lee and Davison. Here according to this model, the plasma membrane is solid in nature. That's why it cannot explain the semi-permeable or selectively permeable nature. Okay, right. Here this is also called sandwich model. Sandwich model means the lipids are sandwiched between the two protein layers, two protein slices. Here just this is the one protein slice and this is the other protein slice. So the two lipids are get sandwiched between the two protein slices. Okay, this is called sandwich model. Second one is unit membrane model. Unit membrane model. Here which was proposed by Robertson. Here according to this model, here the thickness of lipid layer. Here each protein layer is 20 angstroms thickness. Both lipid layers are 35 angstroms thickness. So the thickness of plasma membrane is about 75 angstroms. Okay, this is called unit membrane model. This model also not accepted. Why? Because different biological membranes are differ in their composition and differ in their thickness. That's why unit membrane model also refused or not accepted. Right? Let us see. Well or widely accepted model of plasma membrane is fluid mosaic model. Fluid mosaic model is widely accepted model of plasma membrane. Fluid mosaic model of plasma membrane. Here which was proposed by Singer and Nicholson. Singer and Nicholson proposed well accepted model of plasma membrane in 1972. Their model is called as the fluid mosaic model. Okay, here according to this model, here the lipid molecules are in semi-fluid nature. The proteins nothing but solid, those are icebergs. Okay, here the protein ice pieces, the ice of protein, icebergs of proteins, which is free float on the lipid ocean. This is called as the fluid mosaic model. Okay, according to this model, the fluidity belongs to lipid layer. Lipid layer shows semi-fluid nature. Here the proteins are get arranged mosaic pattern. Mosaic pattern nothing but irregular pattern. So according to this model, the proteins are icebergs which are free float on the lipid ocean, which was proposed by Singer and Nicholson. Here this model explains the lateral movement of proteins. Lateral movement of proteins but not lateral movement of the lipids. Remember, it is very important. Okay, here right. Fluid mosaic model of plasma membrane explains various functions of the plasma membrane. What are the various functions? Cell growth, endocytosis. What is mean by endocytosis? Endocytosis, to bring the molecules into the cells, which is called as the endocytosis. Secretion. Next, intercellular junctions. Intercellular junctions are formed by plasma membrane, not intracellular junctions. Intercellular junctions are formed by plasma membrane. So what are the functions of plasma membrane? Cell growth. Okay, next secretion, formation of intercellular junctions. Next, endocytosis. What is endocytosis? Just I told, endo means inside. So to bring the molecules inside the cell. Here another function of the plasma membrane is transportation of materials into the cell or out of the cell. Here now, why? Because the plasma membrane contains proteins, carrier proteins. Those carrier proteins are two types are there, pumps and porins are there. Here now, the plasma membrane is especially involved in the transport. Okay, here now one is the facilitated diffusion. Facilitated diffusion. Second one is the active transport. Here what is facilitated diffusion? Here generally this is the plasma membrane. Here if any substance which is enters into the plasma membrane, it should dissolved in the lipid layer. Those substances are called as the lipidophilic substances. Lipidophilic substances easily move across the plasma membrane without requirement of carrier proteins. Okay, here right. Now here, what is the condition for transportation of materials across the plasma membrane? The condition for material transportation of materials across the plasma membrane is solubility. The substances compulsory soluble in the lipids. Lipid soluble substances are called lipidophilic substances, nothing but hydrophobic substances. So the hydrophobic substances, nothing but lipidophilic, nothing but non-polar substances can move easily across the plasma membrane. Second one is the polar substances. Polar substances, nothing but hydrophilic substances, means the substances that are dissolved in the water, those are called as the hydrophilic substances. These polar or hydrophilic substances cannot move easily across the plasma membrane. That's why they requires the help of carrier proteins. Those carrier proteins are involved in either facilitated diffusion or either active transportation. What is the facilitated diffusion? The transportation of materials across the plasma membrane from its higher concentration region to lower concentration region along the concentration gradient without requirement of metabolic energy is called as the facilitated diffusion. What is facilitated diffusion? So the movement is take place from high concentration to low concentration gradient. Along the concentration gradient without requirement of metabolic energy, which is called as the facilitated diffusion, or also called passive transportation. Second one is the active transport. Active transport. Here it is quite opposite to facilitated diffusion, means it is some of carrier proteins involved in the transportation of materials from its low concentration region to high concentration region against of concentration gradient, against of concentration gradient. Okay, with the requirement of metabolic energy, which is called as the active transportation. The carrier proteins which are involved in the facilitated diffusion are called as the porins, and the carrier proteins involved in the active transport is called as the pumps. Here next, the water molecules. Here the water molecules moves across the plasma membrane easily. So that is called the movement of water molecule from its higher concentration region to lower concentration region across the plasma membrane is called as the osmosis. What is mean by osmosis? Osmosis is a type of diffusion. It is simple diffusion without taking off any carrier molecules or any carrier proteins. It's called as the simple diffusion. Okay, which substance requires? Which substances requires the help of carrier proteins? Non-polar substances or polar substances? What is the correct answer? Polar substances requires the help of carrier proteins. Okay, right. What is the best example for active transportation? Na plus and K plus pump is a best example for active transportation. Right. Now let us see endomembrane system. Endomembrane system. Here generally the cytoplasm contains various cell organelles. Different types of cell organelles are present in the cytoplasm. Right here, those cell organelles are double membrane bound cell organelles, single membrane bound cell organelles. Here all cell organelles are differ in their functions or differ in their structures. But the endomembrane system includes somewhat cell organelles. Here, those cell organelles are interconnected with each other in their functions. So, the functionally correlated cell organelles, the functionally interconnected cell organelles are included under the endomembrane system. Okay, here, which cell organelles are included under the endomembrane system? Number one, endoplasmic reticulum, ER. Second one, Golgi apparatus or Golgi bodies. Third one, lysosomes. Fourth one, vacuole. All these are included under endomembrane system. Okay, here. Now, I am going to discuss one statement here. All cell organelles that are included under endomembrane system are absolutely single membrane bound cell organelles. This is the first statement. Second statement are all single membrane bound cell organelles are belongs to endomembrane system. Right? First statement is all cell organelles in the endomembrane system are single membrane bound. Second statement, all single membrane bound cell organelles are belongs to endomembrane system. Here, which one is correct? Means only first statement is only correct. The second statement is absolutely wrong. Why? Because all single membrane bound cell organelles are not a part of endomembrane system. Why? Because peroxisomes are not belongs to the endomembrane system. Glyoxysomes are not belongs to endomembrane system. Okay, so all single membrane bound cell organelles are not belongs to the endomembrane system, but the endomembrane system includes single membrane bound cell organelles. Okay, right. Next, here the mitochondria and chloroplasts are not a part of endomembrane system. Why? Because their functions are not coordinated with each other. Their functions are different. Mitochondria is catabolic cell organelle, while chloroplast is anabolic cell organelle. That's why their functions are not interconnected with each other. Hence, the chloroplast and mitochondria are not a part of endomembrane system. Here, what are the parts of endomembrane system? Which cell organelles are included under the endomembrane system? Here clearly discussed: endoplasmic reticulum, Golgi apparatus, lysosomes and vacuoles. All these are belongs to endomembrane system. Okay, why? Because their functions are coordinated with each other. Okay, right. Second one, let us discuss about endoplasmic reticulum. Endoplasmic reticulum. Here, endoplasmic reticulum is considered as system of membrane. It is considered as system of membrane. Second one, here the cell organelles which are arises from the another cell organelle is endomembrane system. Why? Because endoplasmic reticulum is involved in the synthesis of Golgi apparatus, lysosomes, like that. So the cell organelles that are formed from the another cell organelle is endoplasmic reticulum, or the endoplasmic reticulum is a cell organelle which is responsible for production of another cell organelle. So endoplasmic reticulum is cell organelle which is responsible for production of another cell organelles. Right. Here, endoplasmic reticulum, it was first described by K.R. Porter. Here, K.R. Porter observed thread-like lipoproteinaceous structures in the nucleus, and these thread-like lipoproteinaceous structures extend in the cytoplasm irregularly and coined the term endoplasmic reticulum. A network of lipoproteinaceous structures irregularly scattered in the cytoplasm is called as the endoplasmic reticulum. Here, the endoplasmic reticulum occupies over 60% of cell. That's why endoplasmic reticulum is largest cell organelle. Here, system of membrane is endoplasmic reticulum. The cell organelle responsible for production of other cell organelle also endoplasmic reticulum. Largest cell organelle also endoplasmic reticulum. Okay, next, the endoplasmic reticulum is involved, especially involved in the intracellular transportation, but not intercellular transportation. The cell organelle mainly responsible for intracellular transportation is endoplasmic reticulum. Okay, right. The endoplasmic reticulum, for suppose just this is the cell. Here it is the nucleus. Just this is the nucleus, outer membrane and inner membrane. Okay, here now this is the endoplasmic reticulum. Here the endoplasmic reticulum is extends from nuclear envelope to plasma membrane. This is the endoplasmic reticulum. It occupies over 60% of place. That's why it is largest cell organelle. Right. Here now the endoplasmic reticulum here divides intracellular space into two components, but not intercellular space. Intracellular space. Endoplasmic reticulum divides intracellular space into two components. Number one, luminal side. Second one is the extraluminal side. Luminal side means towards inside, means towards nucleus. Extraluminal side means outside of endoplasmic reticulum, means towards the cytoplasm. Right. Here generally the endoplasmic reticulum is two types. Number one, RER, rough endoplasmic reticulum. Second one is SER, smooth endoplasmic reticulum. Right. Here just this is the here rough endoplasmic reticulum. Here this is the rough endoplasmic reticulum. Rough endoplasmic reticulum is due to presence of ribosomes. Ribosomes. Here, which type of ribosomes are found on the rough endoplasmic reticulum means 80s type of ribosomes are present on the rough endoplasmic reticulum. Rough endoplasmic reticulum is meant for protein synthesis. Protein synthesis. Second one is the smooth endoplasmic reticulum. The surface of endoplasmic reticulum is smooth due to lack of ribosomes. That's why it is called as a granular endoplasmic reticulum. Rough endoplasmic reticulum is called as granular endoplasmic reticulum due to association of ribosomes. These ribosomes are associated with the rough endoplasmic reticulum by means of riboporins. So the surface of endoplasmic reticulum is rough due to presence of ribosome. Second one, smooth endoplasmic reticulum. The surface of endoplasmic reticulum is lack of riboporins. That's why the ribosomes are absent on the surface of endoplasmic reticulum. Hence, it is called as the smooth endoplasmic reticulum, or also called as a granular endoplasmic reticulum. Right. I'm going to ask one question. Right. Here, X and Y, two cell organelles. X and Y are the two cell organelles. X is divided into two types based on the Y. The Y is meant for protein synthesis. What are the X and Y? Here, listen my question carefully. X and Y, these are the two cell organelles. X is divided into two types based on the Y, but Y is meant for protein synthesis. What is the X and Y? X is endoplasmic reticulum, Y is ribosomes. Based on the Y, X is divided into rough endoplasmic reticulum and smooth endoplasmic reticulum. So the X and Y are respectively endoplasmic reticulum and ribosomes. Here, what are the functions of rough endoplasmic reticulum and smooth endoplasmic reticulum? Here, the rough endoplasmic reticulum is generally originated from nuclear envelope. Rough endoplasmic reticulum is generally originated from nuclear envelope. Here, it is extend from outer nuclear envelope to plasma membrane. Rough endoplasmic reticulum is concerned with protein synthesis. Next, smooth endoplasmic reticulum. Smooth endoplasmic reticulum is concerned with lipid synthesis and detoxification of substances. What is detoxification? Removal of toxic substances such as pollutants, carcinogens, and other drugs also. So, detoxification is carried out by smooth endoplasmic reticulum, especially in the animal cell. Especially in the animal cell. Here, the smooth endoplasmic reticulum is responsible for lipid synthesis. Lipid synthesis. What are the two lipid synthesis? And especially in the animal cell, it is responsible for synthesis of steroidal hormones such as testosterone, estrogen, and progesterone. All these are produced from the smooth endoplasmic reticulum. Okay, this is about endoplasmic reticulum. Next, about Golgi apparatus. Right. Golgi apparatus. Here Camillo Golgi describes a densely strained structure in the nucleus and reported them as Golgi apparatus. Golgi apparatus is called middleman of cell, director of macromolecular traffic. Next, it is also called lipochondria. It is also called idiosomes. It is also called Dalton bodies or Dalton complex. It is also called Baker's bodies. And it is also called bifacial cell organelle. Bifacial cell organelle. It is also called secretory cell organelle. secretory cell organelle. Okay, right. Here all these are the names of Golgi apparatus. Here generally the Golgi apparatus is composed of cisternae, vesicles and tubules. Here why it is called as a bifacial cell organelles because it has two phases, cis phase and trans phase. Here just this is the cisternae of Golgi apparatus. Here now this is convex phase or cis phase or also called formating phase. Here now this is called trans or concave or maturated phase. So two phases are there. That's why Golgi apparatus is called as bifacial cell organelle. It is also called polarized cell organelle. Polarized cell organelle. Here, Camillo Golgi first of all described the densely strained structures near the nucleus and named them as Golgi apparatus. Right here, the Golgi apparatus is mainly involved in the what are the function of Golgi apparatus? Tra Transportation of materials, intracellular transportation of materials, packaging of materials. Here, during the packaging of materials, first of all, the materials are transported from endoplasmic reticulum in the form of vesicles. Those are first associated with the cis phase. Here, what is the sequence of events of packaging of materials? First of all, endoplasmic reticulum, packaging of materials from endoplasmic reticulum in the form of vesicles, and those are fused with the cis phase of Golgi apparatus, and modified in cisternae, and those are enters exit through trans phase to target site. These are target site. These are the sequence events of packaging of materials through Golgi apparatus. Here the materials that are packed in the form of vesicles from endoplasmic reticulum. First of all, those are fused with the cis phase, and those are enters into middle of the cisternae and modified in the cisternae and exit through the trans phase to the target site. Okay, here this explains that the Golgi apparatus is closely, the endoplasmic reticulum is closely associated with the Golgi apparatus. Like the materials, same here the proteins that are synthesis from the rough endoplasmic reticulum. Here those are enters into the cisternae through the cis phase and modified in the cisternae and exit through the trans phase of the Golgi apparatus. Right. Here, what are the another functions of the Golgi apparatus? Golgi apparatus plays an important role in cell plate formation. The cell plate develops into middle lamella. So, it is mainly involved in the cytokinetics of cell division. Next, it is secretory cell organelles. Why be cell organelle? Why? Because it secretes somewhat secondary cell wall materials such as lignin, suberin, like that. These cell wall materials are secreted from the Golgi apparatus. Hence, it is called as the secretory cell organelle. Next, Golgi apparatus is mainly associated with glycoprotein synthesis of glycoproteins and glycolipids. Golgi apparatus is mainly associated with the synthesis of glycoproteins and glycolipids. What is glycoprotein? The protein which is associated with glucose molecule is called a glycoprotein. Synthesis of glycoprotein is called glycosylation. Synthesis of glycoprotein is called glycosylation. Next, glycolipids. The lipid which is associated with glucose is called as the glycolipid. The synthesis of glycolipid is called as the glycosylation. What is glycosylation and glycosylation? Glycosylation means synthesis of glycolipids. Glycosylation means synthesis of glycolipids called glycosylation. Glycoproteins called glycosylation. This is about the Golgi apparatus. It is called polarized cell organelle. Why? Because here the substances are packed from one polar end to another polar end. That's why it is called the polarized cell organelle. Okay, right. This is about Golgi apparatus. Okay, here in our next video or in our next class, we would like to discuss about lysosomes, vacuoles, mitochondria, chloroplast, like those structures are discussed in our next class. Okay, right. Uh, Bye children. Have a nice day. Okay, thank you for watching video. See you tomorrow in our next class. Bye.
ఇంకెవరైనా ఉన్నా ట్రై చేయండి.
చేస్తాను సార్. రేపు సేమ్ టైం మళ్ళీ
రేపు అదే టైమింగ్ ఉండొచ్చు. నేను ఒకసారి మీకు ఇన్ఫార్మ్ చేస్తాను. రైట్.